Good morning, everybody in the room and everywhere. So this is one of the results of the Positive Atlantic project under the collaboration of the group of environmental biology of the University of Vigo in collaboration with the University of Santiago de Compostela and the group of Xuluma-Cide. I'm going to present during the first part of this presentation a kind of a resume of several ideas that we gathered in these 2 days from the different talks, and at the end, the results of our work. Well, the intense global trade of goods during the last century has promoted an important increase in the introduction and establishment of vespids outside their native range. These insects are successful invasive alien species with consequences for ecosystem dynamics and people throughout the globe. It is the case of Vespula species invading several continents and countries of the northern and southern hemispheres, indicated here in the map that Professor Jacqueline Bex showed us yesterday. And these species are considered some of the most dangerous invasive species, and as she named them, they are super invaders. Despite this condition, between the records related to the invasion of the 3 invasive genus of Vespidae, Vespula works are less represented in the scientific literature than the number of publications of other invasive vespids, such as Polistes or Vespa. Between them, the 22% of the scientific literature deals with several aspects of the biology of Vespa velutina and the implication of its invasion, which is the species that bring us together to this conference. Since the first records of this species in 2003 in South Korea and 2004 in France, it expanded to neighboring regions, invading several countries. In Galicia, the first 2 nests were recorded in 2012, and in 6 years, the quantity of nests increased to more than 26,000, expanding through the whole territory. According to the plot shown by Peter some talks ago, the window to eradicate this species was very, very short, was very small. We had 2 years practically to be able to eradicate it for a short period. Now, at this point, we only have to work on diminishing its effects. This impressive population growth is due in part because it found suitable environmental conditions for its establishment and growth. But it is also related to some biological traits that share with other vespids that enhance its invasiveness capacity. These are, for instance, eusociality, involving task repartition between the individuals of the colony, defense, as we saw yesterday in the Professor Nie conference, the ability to communicate different aspects of the environment, of the state of the colony, and efficiency in resource provisioning. Because they behave as a super individual, all aspects of the biology of this colony occur simultaneously, making them very efficient. They also have an annual cycle, meaning that all the biological cycle is fulfilled in 1 year, and they have polyandry. This means that foundresses queens fecundated by several males search for protected places to hibernate. These make them ideal propagules to achieve distant places with a minimum number of individuals able to produce a new population. In this case, just one individual can bring us a whole invasion on an entire region. Besides human transport, they also have an efficient natural dispersal ability, and queens are able to fly several kilometers far from the original nest. They also have a high reproductive rate, and some studies have revealed that one colony is able to produce more than 3,000 new queens and 900 males. Other relevant biological trait is their opportunist and generalist use of resources that include water, fiber, carbohydrates, here feeding on floral nectar, here consuming sap or fruit, and protein that is obtained by scavenging and hunting other arthropods. The major impacts are expected on related species, such as species of the genus Vespa, here facing one another with Vespa crowro native to Europe, mainly when competing for resources, or as in the case of Vespa velutina invasive in Japan, where males fecundate queens of the native species, producing reproductive interference. Besides competition for resources, some of the most important ecological effects of invasive vespids are related to their predatory behavior, as in the case of Vespula in New Zealand or in Hawaii. There, native trees produce sugary drops of honeydew that it is exploited before the arrival of these invaders. It was mainly exploited by insects, birds, and make an important part of the soil dynamics. However, an abundant population of invasive wasps consuming honeydew reduced this resource, making inaccessible for other organisms, such as birds or these other organisms, thus changing forest dynamics, including as well the pollination of the plants, as Hannah et al. And collaborators showed us in this plant, Metrosideros polymorpha in Hawaii. Then, predation of wasps and hornets at honeybee colonies are important, and change several aspects of beekeeping and pollination dynamics. Okay, as I said some slides ago, vespids are opportunistic foragers. They are very good at finding a source, which is commonly is able in the space and the time, and exploiting it. For instance, they take profit from the attractive strategies of the plants to ensure visits by insect pollination, as the flowers. This is the case of Vespa velutina hunting pollinators on flowers of native plants in Europe. We studied the effects of Vespa velutina for the floral visiting behavior of pollinators and the consequences for the pollination of this plant. This is a common herb visited by a diversity of insects that forage for nectar and for pollen in high summer where there are fewer blooming plants. So it is an important resource for many insects. In this table, we present the results that we published in 2020 with Professor Maria Calvino, in which we have here the main groups of pollinators and here the main variables that are suffering because of the predation of Vespa velutina on the plant. we found significant changes in the frequency of visits by honeybees, these red squares, and other bees too, flower visitation rate of Apide, as well as a reduction in the time that bumblebees and syrphids, Diptera syrphids, spend visiting the flowers. This means that the hunting pressure of the hornet in the flowers changes the two dimensions of the pollination effectiveness of the plant. On one side, when the insects pay shorter visits to the flowers, this reduces the qualitative component of these insects as pollinators, reducing the quality of them as pollinators, and the diminishing in the number of visits is detrimental for the quantitative component of pollination effectiveness. As a result, we observed a reduction in the number of pollen grains deposited on stigmas of the flowers of the plants where velutina was hunting. This is one piece of evidence that reflects how Vespa velutina affects ecosystems dynamics in the invaded areas. Currently, we work together with a group of Juliet Osborne of the University of Exeter, and we continue studying the effects of this invasion for pollination services. However, one of the most evident effects is indeed its effect as a predator of honeybees. These are intensively predated in front of the hives along the day, during the summer and autumn, during practically all daylight hours. First, bees are able to display some defensive strategies, as this small bee carpet we see here. But afterwards, when these defenses are ineffective and they continue being predated, they end staying inside the hive. They don't go for foraging, and this phenomenon has been called the foraging paralysis, meaning that workers do not go for collect nectar, pollen, food. Thus, and states in this path model that Dr. Fabrice Requier showed us yesterday, the hunting hornets impact negatively the honey reserves in the red arrows, honey reserves, but particularly they affect the colony survival. The researchers also found that the hive muzzle, this device in front of the hives, helps honeybees to reduce the negative effects of predation by reducing the foraging paralysis. However, with this method, hornets are not being captured, that it is possible that they continue hunting bees and other insects in other locations, as we already have seen they do it in flowers. So it is of paramount importance to continue exploring and scientifically validating the methods to protect pollinators, including honeybees from the predation of Vespa velutina. Then, one of the most used methods by beekeepers in Galicia are the electric harps. We already saw one of the effects of electric harps in Damian's presentation, and I'm going to explain other details of the use of this device. This, as he said, consists in a frame with vertical parallel wires that produce an electric charge of different poles. When hornets fly in front of the hives to hunt honeybees, they touch both poles, receiving an electric charge, then stay paralyzed for some moments, then fall into a cage placed downwards, and finally where this has a mesh that allows smaller insects to escape, while the hornet crawls inside after recovering and finally ends into a bottle where dies. The objectives of our work were to assess the effectiveness of electric arcs to protect honeybees and compare the performance of colonies that are protected with these devices against colonies unprotected. To achieve these objectives, we studied 36 colonies placed in 3 apiaries in northwest Spain at the coastal region of Galicia. We had 12 hives per apiary divided into 2 groups or 2 treatments. One line of hives were protected with electric arcs, as we see here. And the other 6 hives were unprotected. Then Vespa velutina was able to hunt honeybees at libitum. Then, from June to October, we made monthly assessments of the quantity of hornets hunting in front of the hives, the quantity of predated bees, and also made a complete monitoring of the colony, including the quantity of foragers, workers inside the colony, pollen income, brood, pollen stored, quantity of uncapped and capped honey. And also in October, we collected and weighted random samples of 30 workers of each colony to weigh them and compare them. We found that in the apiaries, we had different hunting pressures, different numbers of hornets hunting in front of the hives. This is a rate calculated over 10 minutes. So each panel here, it represents one apiary. One had a high hunting pressure, another an intermediate, and another a low hunting pressure. These are the months where we monitored the variables, and we show here in red the behavior of hornet hunting in the unprotected hives in red, and in blue the protected hives. We found statistical differences between treatments. Thus, hives are an effective method to reduce hunting pressure and predation in hives. We see here higher levels of predation in unprotected hives in the 3 sites. In the site with low hornet numbers of hunters, it was minimum, it was 0. And in general, there was an increase in hunting pressure along the season, with the exception, as Damian said, of September in 2 of the sites. This was probably related to a period very, very warm, approximately 40 days before. Then this heat wave probably affected the abundance of the hornets on September. Okay, so we here see again this plot that Damian showed us some moments ago. Here on the left we have the number of workers going out or into the hive, foragers basically, and during 5 minutes. The red bars are again unprotected hives, and blue bars are those protected hives. These red arrows point to those months where there was an evident protective effect of the hives. We see here that unprotected hives were in foraging paralysis. This means no workers were going out or inside at all during these months in this site. On the contrary, in the blue bar, the hives protected, most of the time there were workers going for food, going to the field for foraging. In the last month, the predation pressure was so high that the electric arcs were not able to control all that charge of hornets. So in this side, the paralysis was total. On the different perspective we observed in the site with intermediate predation pressure, in which occurred a paralysis, but this was not total. And we have to say that it is not only a foraging paralysis. Foraging meaning that the animals go searching for food or water. Actually, bees stop making many or practically all the processes that they make outside the hive, including fanning to cool the brood, bringing resins for making propolis, which is a very important process into the hive, and very important for the colony health, they stopped doing hygienic behaviors, such as cleaning the hive for feces, carcasses, or sick individuals. Thus, we hypothesize that Hagley attacked colonies are more prone to suffer higher levels of parasites and sickness. The paralysis was also evident in the numbers of workers inside the hive, as we see here in this plot. When observing the quantity of workers inside the hives, we saw this. It could be at first glance, it can give the idea of that attacked hives have more individuals. But this is not like that. This means that all the workers that should be in the field, they are not outside. They are inside the hive consuming honey, and it is not a big surprise that honey and pollen stores are also significantly diminished in paralyzed colonies. On top of that, the fact that attacked colonies have no workers foraging, this is totally reflected in the quantity of pollen income. As we see here in the total paralyzed colonies, there is no pollen income. The problem is that pollen is one of the basic resources for feeding the brood. Thus, as a consequence, the quantity of brood that we see here was significantly reduced in those highly attacked hives, in comparison with protected hives with electric arcs. This will have a very important impact on the quantity of workers that will keep the colony alive during the last part of the summer, the autumn, and particularly during the winter. Overwintering workers are also responsible to initiate, not only to make the colony survive throughout the winter, but also the activation of the colony in spring. This means that the colonies under hunting pressure are impoverished in number of individuals in overwintering workers. But this is not the only effect we saw of all this debilitation process of the colonies triggered by the predation of Vespa velutina. What's even worse, we have found that here, for the first time, we found evidence to conclude that the workers in unprotected hives have less weight than those of protected hives, which had better levels of resources and health. In this plot, we see here the percentage of survivorship of our experimental hives, in which those unprotected in gray and those protected in black and those protected in gray colonies of the 3 apiaries, high, intermediate, and low. What we see here is, and we confirmed it statistically, is that unprotected hives have a lower survivorship. It was 30% lower in the site with high hunting pressure and 60% lower in the site with intermediate predation pressure. This was a little a surprise. But we have found that this difference is related to the environment. Since in this site, which is found at a higher altitude, has a shorter blooming period and a harsher winter, less sun and more cold days. Thus, together with the hunting pressure, the site and the availability of resources can also play an important role to determine the survivorship of the honeybee colonies that are being attacked in the invaded areas. Thus, it is more complex than only measuring and controlling one control method. It is necessary to see the environment to the site too. That component is very important as well. In resume, in all cases, what we demonstrated with this work is that colonies under hunting pressure have less individuals, lighter overwintering workers, and scarce resources. This induces higher deaths of colonies in the winter and beginning of the spring. As a conclusion, we have that high and intermediate predation affects the behavior of honeybees and the survivorship of the colonies. The hunting pressure causes decreases in foraging activity, pollen and honey income and stores, number of individuals of different ages, and the weight of overwintering workers. We concluded also that electric hubs help to reduce predation pressure and paralysis. Here we show, as an hypothesis, based on what we know so far of the process triggered by the predation of this invasive hornet, including our work and studies showing this schema, where predation diminishes the number of workers and produce changes in the behavior of bees. This affects the collection and storage of resources that, together with the stress, influence the weight of overwintering hornet workers and increase the susceptibility to suffer diseases. All these parameters together produce a higher mortality of colonies that reduce the profitability and sustainability of beekeeping. And also, when diminishing the number of colonies and a reduction of floral visitors, this probably affect pollination services. Some recommendations for the management of apiaries in invaded areas thus are: first, setting electric arcs perpendicularly to a line of hives to help to reduce hunting pressure and paralysis. Second, in apiaries with high hunting pressure, additional methods such as nest destruction are very, very recommendable. Then, reinforcing colonies by merging them before wintering so they have more individuals and are stronger and then can survive better. Also, facilitating the access of bees to the diverse floral resources and fresh water sources to reduce homing failure. And finally, it is important also to consider in highly invaded areas a supplementation of a diverse diet rich in different nutrients, not only in winter, but also when they suffer partial or total paralysis, trying to avoid the production of smaller and weaker workers with the aim to reduce the deaths of the colonies that are being under attack. Finally, I want to say thank you for the participants in this Congress. Thank you to the organizations and organizers. Thank you to all partners of this Atlantic Positive project, and to the different people that have supported our work. Now it's time for questions and comments.